Hello. I am putting this video together briefly in the possibility that we run into some time crunches in my biology class, and I want to make sure that I address everything that's in your end of semester assignment. The one topic I want to cover today is something called DiHybrid Cross. And so this video is going to focus on that. It's going to assume you already understand simple dominance with only two alleles. Simple dominance, two alleles, single trait problems. So that means like big B, little b. Big B is dominant to little b. Big B stands for some dominant allele. And in the hybrid, whatever phenotype is associated with the big B allele will show in the organism. So that's what I mean when I say monohybrid or one trait, two allele problems. Dihybrid is really just looking at two traits. And in this case, we're looking at two traits that are each simple dominance. So let me switch over to the camera here and show you what I'm talking about. So we've studied problems like this. Let's say that we were doing, I don't know, eye color. And there were only two eye colors possible, which we know is not realistic, but whatever. Brown eyes and blue eyes. And let's assume that brown eyes are due to a gene that we'll label big B. We'll say that's the big B allele. And that gene is dominant to the allele, the gene that produces blue eyes. So we're going to use these symbols for those letters. And that means if we were to make a chart that lists all possible genotypes for this one trait, eye color, you could have an individual who has only brown genes, in which case their phenotype would be brown eyes. Likewise, because the big B is dominant, they could get the brown allele from one parent and then inherit from the other parent maybe the blue allele. But because brown is completely dominant to blue here, we're not going to see any codominant or incomplete dominant behavior. We're going to see absolute dominance. It means that this person with this genotype, their eyes will be just as brown as those. There's no distinction between the two. When you look at a brown-eyed person, if this was the genes, they aren't that way in reality, but if these are the genes, both of these people would have equally brown eyes. There'd be no evidence in their phenotype that they carried a different genotype from each other. The last type of available genotype is, of course, little b, little b. Remember, we write little b, big B the same as big B, little b. So we won't be writing any genotypes down like that. But this hybrid form does cover the idea that they get the little b before they get the big B somehow. At any rate, little b, that's a little b from one parent, a little b from the other parent. That's going to produce an organism with blue eyes. All of that's reviewed fairly easy. Now let's assume that there's a different characteristic on this person, another feature of their biology in which, I don't know, let's say it is whether their hair is curly or straight. And let's suppose that straight hair is dominant to curly hair. Now, right here, I'm going to point out something. A capital S and a lowercase s look a lot alike, and a capital C and a lowercase c look a lot alike. So I'm going to use the letter H here. Big H is the gene for straight hair. It is purely dominant to little h, and there are no other genes in this example. If we make a GP chart for these, big H would produce the straight hair phenotype. Big H, little h would produce the straight hair phenotype, and little h would produce the curly hair phenotype. So all of this should be plainly evident from stuff we've covered already. Now, here's where things get interesting. What if we want to write down the genotype of a person who's hybrid with brown eyes and hybrid with straight hair? Well, if you want to write down their entire genotype, you're going to have to write all the genes they have for this trait and all the genes they have for this trait. So it looks a little weird. And what we're going to do is we're just going to, while we're working this problem, just agree to always put the eye color genes first, followed by the hair color genes. So if they were hybrid with brown eyes, they would have big B, little b for this first trait. And they would have, if they were hybrid for hair, curly hair, straight hair, they would have big H, little h for their hair genes. Notice also, this is one of the reasons that we always use the same letter for any one trait. So B's are going to use for all of our eye color genes here, and H's are what we're going to use. This makes it easy to keep all of our eye color genes grouped together and all of our hair texture genes grouped together. So this would be somebody who is hybrid for both traits. We call that condition dihybrid. It's pretty easy. Hybrid for two traits, hybrid for both traits. Now, the question that we want to answer today, what this lesson is really about, is what happens if you take an organism, a person, that is hybrid for two traits, each trait being simple dominance, and you make them with another person who is hybrid for those same two traits. What kind of offspring could they produce? And there are two approaches to this. One approach is approach it where you're looking at all of the genes at the same time. Now, before I go forward, I want to point out something really important. These genes here, these two letters, represent a homologous pair. And when a person sexually reproduces, the first thing they do is make gametes through the process of meiosis. And meiosis will choose one of this homologous pair to put into the gamete. But it does that for every homologous pair. So this is a second homologous pair here. So what this means is, when we're doing our Punnett square, which basically illustrates meiosis and fertilization, this organism, big B, little b, big H, little h, they could produce a gamete that has one of these, let's say it's big B in this example, and one of these, say it's big H. This is one of the four kinds of gametes this organism could produce, because one from this homologous pair and one from this homologous pair. Another way that I've explained this whole idea before is for every trait you get, it's going to be on a chromosome, and there's going to be another chromosome in there that also carries genes for that trait. We call those two chromosomes the homologous pair. One homologue comes from one parent, and the other member of the homologous pair comes from the other parent. So this one might be from the mommy of this guy, and this one might be from the daddy of this guy. But the two together, this is on one chromosome, this one's on another chromosome. They make up homologous pair. One came from dad, one came from mom. The other homologous pair is a separate pair of chromosomes. So you can think of each homologous pair like a coin, where the genes on that chromosome that you got from mom are on the head of the coin, and the genes you got from dad on that chromosome are on the tail of the coin. And when you make a gamete, when you produce a sperm or an egg by meiosis, it's as if you flipped that coin and it landed, and whichever face is up, that's the chromosome that'll go into the sperm or egg you're making. But here's the thing. You, as a human being, have 23 homologous pairs. That means you got a coin with the B's on it, and you got a separate coin with the H's on it, and another one with your your skin color, and another one with the shape of your earlobes, and on and on and on for all the different proteins your body might make. So you have 23 such coins. Each coin has the chromosome from your mom on one side and your dad on the other. And every time you make gametes, it's like you flipped all 23 of them, took whatever was face up, and put those chromosomes into that egg or sperm. And then you pick them all up and you flip them again for your next egg or sperm. You pick them all up and you flip them again for your next. So each of them can flip either way. So what that means here is if this is one homologous pair and this is the other homologous pair, then it's sort of like you could get heads and heads, heads on the first coin and heads on the second coin. That would produce this. Your next gamete, though, could be heads on the first coin and tails on the second. Big B little h, one of each homologous pair. Likewise, we come on down and we go tails on the first and heads on the second. That would be a little b big H gamete could be made. And lastly, you get tails on both, and that will produce a little b little h. So an organism with this genotype can produce 4 unique kinds of gametes for that trait, for those 2 traits, actually, 2 traits, hair and eyes. Now, if the mate of this one has this genotype, then they would have the same genotype here, and in fact could produce sperm that are big B for the first trait and big H for the second. Big B for the first trait and little h for the second. Little b for the first trait and big H for the second. And little b, little h. So these are the kinds of sperm that could be made. And then from here, you fill this out normally. Now, what's going to happen here is I'm going to speed up the video during this next section as I fill this out, because I feel like this is pretty obvious how this would go. So, big B, big B. Big B from dad, from mom, and a big B from dad. And big H from mom, and big H from dad. Purebred dominant. Big B, big H, little h. Big B, little b, big H, big H, big B, little b, big H, little h. Now, what's interesting is if you look at this cell, you'll find it's the same as this one, because we have a big going here and a big little going here. We have a big going here and a big little going here. So these two are going to be the same. You get this diagonal symmetry is what I call it going on. Same thing would be true here. You get big B, little b, big H, big H. And then down here, you get big B, big H, little h. I'm going to skip that cell for now and do these other 2 diagonals, because what's kind of cool here is big B, big H, little h, and big B, big H, little h. Notice all of these are all dihybrids. That's kind of cool that we made it a dihybrid, and all the diagonals come out dihybrids. These are not dihybrids. This is purebred. This is purebred hybrid. This is, oops, I left out a letter here. Got to be careful there. Big B, big H, little h. This is purebred hybrid. This one's hybrid purebred. This one's hybrid purebred. You get the idea. One thing I want to do right now is stop. And you'll notice that phenotypes, if we ignore genotypes for a minute, these are going to be dominant for both traits. But this one's going to actually be, the phenotype will be dominant on both traits, because this is dominant up here and this is dominant up here. So this one is also brown-eyed and straight-haired, as is this one, as is this one. All of these, all 9 of these, there are 9 of these, are dominant phenotype on the first trait and dominant phenotype on the second trait. These 9. Interesting. There will be no others that are like that. Likewise, here, we're going to get big B, little h, little h. Now, that one's interesting because they're dominant on the first trait in their phenotype, but they're recessive on their second trait. And there are 2 more that are like this. This one down here, it's a big B from mom and a little b from dad, but they're still dominant phenotype here, but 2 little h's recessive on both. So dominant on the first trait and recessive on the second. And because of the diagonal symmetry, you would see that this one is the same genotype, which means we have 3 that are dominant on the first trait, that is brown, and recessive on the second trait, that is straight. Brown eyes and straight hair. Dominant, recessive. This one would be brown eyes. I'm sorry, this isn't straight hair. I lied. They're recessive. Recessive hair is curly. So they're brown-eyed, curly-haired. This one's brown-eyed, straight-haired. Now, these next 3 here, you're going to find, if you do the work, little b, big H, little b, big H, little b, big H, little h. These 3 are all recessive on the first trait, but dominant on the second. In other words, these are blue-eyed, straight-haired. And there's only one that gets little genes from both parents for both traits. One that is recessive. In other words, this is the only blue-eyed, curly-haired offspring from these two parents. So this is a pretty big Punnett square, 16 squares. 9 plus 3 plus 3 plus 1 is 16. In other words, another way to say this is 9/16 of the offspring will have brown eyes and straight hair, slightly over half. Okay. Here, 3/16 will be like this, 3/16 will be like this, and 1/16 will be like that. That's the first way to do the dihybrid cross, and it's fairly straightforward. It really kind of makes sense. These dihybrids here I just kind of mentioned because I thought that was interesting, but they fall into the 9 here that are dominant in their phenotypes. Now realize this is all about phenotypes of the offspring. All about phenotypes of the offspring. I'm not going to do the genotype ratio, and I'll tell you why. It's actually 1 to 2 to 2 to 1 to 4 to 1 to 2 to 2 to 1, which is a crazy ratio. So we are not going to worry about the genotype outcome of the dihybrid cross. All we care about is the phenotype outcome, and if they're both simple dominant traits, it will always be 9-3-3-1. Dominant, 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 recessive, recessive, dominant, recessive, recessive. You see a pattern here. Memorize that pattern, memorize this number, and you'll never have to do one of these stupid Punnett squares again with 16 squares in it. Now, I said there's a second way to look at dihybrid cross, and that is the separate way of looking at dihybrid cross. So you've got that. The other way to do it is say, well, what if we looked at just the eye color stuff, then just the hair texture stuff, and then considered the problem as joint probability, which we covered the other day in class. You might remember the way you calculate joint probability is the probability of one outcome multiplied by the probability of the other outcome gives you the probability of the two happening together. And so if we do that, you'll find that this is actually a very simple problem. We have a big B, big H, little h, crossing with a big B, big H, little h. You've done this Punnett square so many times, you probably don't even have to think about it. You get three-quarters of the offspring show the dominant trait. This is phenotypes only. I'm not going to worry about genotypes in this problem. And 1/4 gets the recessive, right? If you look at just the H's, so big B, little b, big H, little h, crosses with big B, big H, little h. This is the same stupid thing. Big H here, little h there, big H here, little h there. Two big H's, hybrid, recessive. These three-quarters, will show dominant phenotype for this cross, and 1/4 of the offspring will show the recessive for the second trait. So what are the chances that one will come out dominant on the first trait and dominant on the second? Well, the chance of coming out dominant on the first trait is 3/4. And the chance of coming out dominant on the second trait is 3/4. So the chance of coming out dominant on both is the joint probability of the two, and it's 9/16. 9/16, 9/16. It's the same thing. If you treat them as separate traits and joint probability, you get 9/16. Well, wait a minute. Maybe this only applies to this one specific case. Maybe dominant recessive won't be the same as my stupid Punnett square. Well, actually it is, because the chance of being dominant on the first trait is 3 out of 4. The chance of being recessive on the second trait is 1 out of 4. You multiply those together and you get 3/16. Dominant recessive 3/16. Same as the Punnett square. Lastly, well, not lastly, two more to do. Recessive on the first trait, that means blue eyes, and dominant on the second trait, straight hair. Recessive on the first trait is 1/4 of the time. Dominant on the second trait is 3/4 of the time. So multiply this, we get 3/16 of recessive on the first and dominant on the second. 3/16 recessive on the first, dominant on the second. Blue-eyed, straight hair. And then the last one, recessive, and you can do this by now. It's 1/4 on the first one and 1/4 on the second one. Multiply those together. 1 out of 16. 1 out of 16 recessive on both. That's the dihybrid. The answer is always 9331 if you're taking two dihybrids with simple dominants. There's your lesson. So in your end of semester project, you just need to make up a problem that says something along the lines of, my creature, the Dubartosaurus. The Dubartosaurus has a dominant eye color gene for brown and a recessive eye color gene of blue. Similarly, in Dubartosauruses, they can have curly hair or straight hair. Straight hair is due to the dominant allele, big H. If an organism hybrid with for both traits mates with another organism that is hybrid for both traits, what kinds of offspring would be produced in what ratio? That's the question. And then you duplicate either this work here or this work here and say the outcome is 9/16 will be brown-eyed and straight-haired. 3 out of every 16 on average will be brown-eyed with curly hair. 3 out of 16 on average will be blue-eyed with straight hair. And 1 out of 16 on average will be blue-eyed with curly hair. And that's dihybrid. Not too bad, I don't think. Best of luck, and I will save this and upload it shortly.